So when we're talking about our generalized human cell, our animal cell, this is what we're talking about. Everything inside of this cell has a specific function, and these things are called organelles, right? They're those tiny organs. We have our nucleus, which has all the genetic information in it. It has all that DNA. Inside of our nucleus, we have our nucleolus, which is packed full of ribosomes, and ribosomes make proteins. So if we move out here, we see all these little dots throughout the cell. Those are our ribosomes that make proteins all throughout the cell. We also have so talking about ribosomes, we can say we have our rough and smooth endoplasmic reticulum, and our rough ER is covered in ribosomes, so it also makes proteins, okay? Our smooth ER does not have any ribosomes on it. It makes lipids and it makes hormones or steroids. It makes steroids, lipids, hormones sometimes. And those get sent to our Golgi body or our Golgi apparatus, and they are packaged and modified in our Golgi body, okay? So they kind of work in tandem. We have our mitochondria. Our mitochondria make energy in the cell, so they're called the powerhouses of the cell. We have our lysosomes, and our lysosomes are special vesicles that are filled with enzymes that they destroy dead things in the cell, they destroy things that the cell doesn't need anymore. So those are our lysosomes. They're the trash guys of the cell. We have our cytoskeleton, which does the same thing that a regular human skeleton does. Our cytoskeleton gives support, it offers a bit of protection, but mostly it just gives support to that cell. Our cytoplasm is that jelly-like substance that fills all the empty spaces in here. It fills all the empty spaces in that cell, and it allows things to move around in the cell, and some processes take place in that cytoplasm. So in this picture, we have our nucleus and our nucleolus. This is that electron micrograph. So this is under that really high-powered microscope. Our nucleus and nucleolus. We have our centrioles in this picture. So our centrioles build up spindle fibers, and whenever our cells are replicating, those spindle fibers pull chromosomes apart, okay? So that is what our centrioles do. And you can see an electron micrograph of our centrioles in a cell, in an actual cell. Like we said earlier, our rough ER has numerous ribosomes on its surface, so it makes special proteins for the cell. So we know that our rough ER makes those proteins for the cell. And here we have a picture of an electron micrograph of our rough ER. You can see the ribosomes on the surface. So in this picture, we have our smooth ER, so it doesn't have any ribosomes on its surface, and it has a different function than our rough ER. Our smooth ER makes lipids and steroids, and it does a couple other things, but primarily it makes lipids and steroids for the cell. Of course, our ribosomes are tiny organelles made of RNA. They have two subunits, but we won't worry about that now. And they make our proteins, right? It's the site of protein synthesis. They're very important for the cell. Next, we have our Golgi body or our Golgi apparatus. These are stacks of membrane-bound vesicles. So they, remember, they package and modify the products of our endoplasmic reticulum. So when we talk about our cell membrane, we can call it many, many different names. One example is a fluid mosaic model. So that's just referring to its fluidity. It can move, it can change. And it's a mosaic because it has all these bits and pieces stuck in it. It's not just one simple layer. So this is, again, our cell membrane. It's what covers our entire cell. Another name we can call it is that phospholipid bilayer. So what that refers to is these two outer layers of our phosphates, and our inner layers are two inner layers of our lipids. So it's kind of like a sandwich in a way. We have our phosphate bread, and the lipids are the insides, whatever inside you like on a sandwich. So our phosphates, they stick to the outsides because they are hydrophilic. They love water. And our lipid layer, everyone knows when you mix oil and water, they just separate. They don't mix. And that is because oil is hydrophobic. It hates water. But that works for this pair because those lipids stick on the outside, and those tails, those lipid tails, stick to the inside. So there's phosphates on the outside, those lipids on the inside, and they just stick together, and that's how they like to be. And there are two layers, so that's where we get the term bilayer from. So we have our protein channels in here. There are lots and lots of things going on in this cell membrane, but we're mainly going to focus on our protein channels. So in your own words, what is your definition of diffusion? And can you give any examples of how you use or experience diffusion on a daily basis? I know a lot of people were talking about the tea and hot water, perfume, cigarette smoke. We got a lot of really good examples. So in diffusion, if you remember, we're talking about the movement of molecules from a high concentration to a low concentration. And in this picture, in this example, the high concentration here is this dye. In this spot, there is a high concentration of dye. And in the water surrounding it, there is a low concentration of dye. So that's what we're comparing. We're starting off with the high concentration here, and over time, these molecules begin to move and spread out until they reach a state called equilibrium. So equilibrium is where there is no net movement of molecules. Everything is spread out, everything is happy, everything has room to wiggle, basically. And equilibrium happens and diffusion happens because these molecules have their own kinetic energy. So when we're talking about a cell, when we talk about diffusion, those molecules that go through the cell membrane do not use the cell's energy. They are small enough to slip through those phospholipids. So they use their own energy to slip through the cell. They use their own kinetic energy. So, like we just said, diffusion is the movement of molecules from a region of high concentration to low concentration. And when we're talking about those concentrations, we're talking about the concentration of the molecule. So in other words, a molecule is moving down its concentration gradient, which means it's moving from a high to a low concentration gradient. Those molecules have kinetic energy, and that kinetic energy makes them move around randomly. They just bounce around. They just have energy that causes them to bounce around randomly. And this bouncing causes those molecules to spread out, basically. Ultimately, the goal of diffusion is to reach equilibrium, so everything is spread out evenly. So molecules are still moving in and out of the cell, but there's no countable movement. Every time one molecule comes in, one goes out to replace it, and vice versa. So there's no net movement of molecules. So as you can see in this animation, diffusion is a passive process. So it does not use any energy from the cell. The only energy that is being used is that natural kinetic energy inside of those particles, or those molecules, excuse me. And here we can see that on the inside of the cell, we have lots of small molecules and we have a few large molecules. So this animation will show you what will happen when diffusion occurs through this membrane. So what will happen? Those molecules, the small molecules that can move easily on their own, are going to move through that cell membrane from a high concentration to a low concentration. So from a high concentration inside to a low concentration outside. So in this example, in this illustration, we had these small red molecules on this side and none on the outside of the cell. These blue molecules, we'll call them sugar molecules, they are too large to move through this cell membrane. So if this wants to reach equilibrium, these smaller molecules need to go through. So what has happened is these smaller molecules, because they are able to go through the membrane, they have gone through, they've diffused through the membrane using their own energy, not using any energy of the cell. And because they have gone through that membrane, they have now reached equilibrium. This is important in many parts of the body. One really good example is in the lungs. So the blood, when it goes through your lungs, when it goes to your lungs, it swaps carbon dioxide for oxygen, and that gas exchange is done through diffusion. So it's able to happen really quickly and it's very efficient. Okay, so that is diffusion. It's the movement of molecules through that membrane, not using any energy of the cell. It is passive. That means it does not use the cell's energy. So there are 4 factors that determine the rate of diffusion. So the first one is the steepness of the concentration gradient. So if you have more molecules outside of the cell than inside of the cell, those molecules are going to rush into that cell much quicker than if there were less molecules. So it makes sense if you're in a crowded building, if you're in a crowded place, especially now, if there are loads and loads of people in there, whenever the doors open, people are going to be coming out at a much faster rate than if there were not as many people. So the steepness of the concentration gradient makes a big difference on the rate of diffusion. Temperature also is a big influence on the rate of diffusion. So that's why we heat up our water. We boil water before we make tea, because when we put the tea bag in that hot water, those molecules are moving so much faster. Heat is energy. So heating up that water creates more energy and allows those molecules to diffuse faster. Another factor is the surface area. So the smaller a thing is, and this is why cells are so sufficient, because they're so small, they have a huge surface area. So when you're looking at a cell, well, you can't look at it obviously, but the surface of the cell is so big compared to the inside of the cell. So that gives you more area for molecules to go in and out of that cell membrane at any one time. And the fourth one is the type of molecule or the ion diffusing. So large molecules, anything that is large is going to require more energy. So large molecules will need more energy to move, so they can diffuse more slowly. Also, molecules with no charge, so nonpolar molecules with no positive or negative charge, tend to diffuse more easily than polar molecules. And this says it's because they are soluble in the nonpolar phospholipid tails. So they can just sneak through much more easily. So some examples of molecules that diffuse through the cell membrane are oxygen, carbon dioxide, and water. So these molecules are very, very small. They're all very small, and they can move through that membrane very quickly. They kind of slip in between those molecules, which is very useful for us, and it makes our bodies much more efficient. So our first diffusion was diffusion through the membrane, right? So diffusion through that phospholipid bilayer. Now we're going to talk about facilitated diffusion. So this is what happens when those molecules are too large or they are polar and they have a strong charge and they can't just sneak through that membrane. So we have facilitated diffusion, which are large polar molecules like glucose and amino acids. They have to use facilitated diffusion to get across the phospholipid bilayer or to get into the cell. So how they do this is they have to pass through a protein channel. So remember when we were looking at that slide with that fluid mosaic model on it? That model had those protein channels in it, and those protein channels are what is facilitating this diffusion. So they're what is helping, that facilitating, they're helping these molecules to diffuse. So the movement of these molecules is still passive, just like regular diffusion. It's still passive. The only difference is the molecules have to go through that protein channel instead of passing between those phospholipids. So they're using a protein channel that is embedded in the cell membrane to get into or out of that cell. Okay? So you can see here that this is just another example of that fluid mosaic model. So this, you can see that phospholipid bilayer, some cholesterol stuck in there, all sorts of stuff. And you can see that transport protein, so that protein channel that is ready to take in something through facilitated diffusion, which we will talk about in a second. And this animation is just giving you more of a visual idea of what facilitated diffusion is. It is still passive, but these molecules are passing through a channel instead of just through that phospholipid bilayer. So osmosis is a specific type of diffusion. So we've talked about diffusion in a general sense. So diffusion is the movement of molecules from a high to a low concentration through that cell membrane, through that semi-permeable membrane. When we talk about osmosis, oh, and of course, diffusion does not require any energy. It is passive. It's not the cell's energy. It does not require the cell's energy. Osmosis is a type of diffusion. So when we talk about osmosis, it is specifically the movement of water, okay? So osmosis is the diffusion of water from an area of high concentration of water molecules to an area of low concentration of water molecules across a semi-permeable membrane, which is just another word for our cell membrane. So obviously, our cells need water. Our cytoplasm in the cell is 90% water. So our cells need water to function. So osmosis is very important in human bodies, in animal bodies, and in anything that's living. It needs osmosis to occur. So in this example, of osmosis, on one side, inside of the cell, we have a dilute solution. So that's a solution with lots of water and very little other molecules. So that is dilute. And on the outside of that concentration, or at the outside of the cell, we have a concentrated solution. So that means there are more other molecules than there are water molecules, okay? So it's a very low concentration of water molecules. So on the outside of the cell, there is a low water potential. On the inside of the cell, there is a high water potential. There's a high concentration of water molecules. So when we're talking about osmosis, we're talking about the movement of water, right, across that semi-permeable membrane. So which way do you think these water molecules are going to move? Are they going to move inside of the cell or outside of the cell? So this is that intermediate stage. So we have a high concentration of water molecules inside the cell and a low concentration outside. So these water molecules are going to move outside of that cell via osmosis to dilute out these larger molecules. So our water molecules in this example, they have begun to move outside of the cell. They have moved to dilute out to become equal with these sugar molecules. So our sugar molecules cannot move across the membrane. So our water molecules have to move to um, create an equal environment with our sugar molecules. As you can see on the inside, we have two sugar molecules, two large sugar molecules, and 5 small water molecules. So that then creates an equal environment. It creates equilibrium with this side of the membrane. And on the outside, we have 1, 2, 3, 4, 5, 6 sugar molecules and several 2, 4, 6, 8, 10, 11 water molecules. So those molecules create an equal environment. They've created an equal environment on the outside of the cell as well. Now you may look at this and say, well, that's not equal. That doesn't look like it's reached equilibrium. But the water concentration on each side, the water potential on each side is equal. And that is because these sugar molecules cannot move through this membrane. It's just water moving. So it's easier for those water molecules to go back and forth to create an equal environment with these sugar molecules, because these sugar molecules cannot move through that membrane very easily. They're too big and they are polar, so they're staying put. So our water is moving to make up the difference. So as you can see, we have equal parts sugar and water, equal parts sugar and water. And like I just said, so these water molecules are going to try to dilute out molecules that cannot move themselves. So these molecules that are too big or that have a charge, this these water molecules are going to move to dilute them, which makes the job easier, and we don't have to use energy like if we were moving those large molecules sometimes. So jumping back really quick, this is an example of osmosis in our blood cells, okay? So this is a blood cell, a red blood cell. In two examples, this blood cell, in the first example, this blood cell has been placed in distilled water. So it is pure water. What is going to happen is there are more molecules inside of that red blood cell than outside. So in the water surrounding that red blood cell, there are no salts, no sugars, nothing other than water. So water is going to rush into those cells from the solution it's been put in. That distilled water is going to rush into those cells to try to dilute out those molecules inside the cell. And that could cause the cells to swell and burst. So this is called lysis. These cells are bursting. In another sense, we start off with our regular healthy red blood cells. And these red blood cells are placed in a concentrated salt solution. So that salt solution causes the water in that cell to rush out of the cell and leave the cell. And all that's left inside the cell are those molecules that cannot get out. So this is just an example of the slide we just saw. This is really what happens to our red blood cells. So you can see they are starting to shrivel. And this state here, when they're shriveling because of a lack of water, is called crenation. They are crenating. So if you're able, take a second, and if you already know, that is great. But look up what the term isotonic means and what are isotonic drinks. So isotonic is being in a equal state of water inside and outside of the cells. Now look up, have a search, or if you already know, that's fantastic, about what an isotonic drink is, because they are they're used in a very specific circumstance for a very special reason. Isotonic drinks. In some cases in the cell, cells must maintain very high or very low levels of some molecules, and this cannot be done with passive transport. So diffusion, facilitated diffusion, and osmosis cannot maintain very high, or very low levels of some molecules. And that's because they do not use cellular energy. Remember, in diffusion, it's always going from a high concentration to a low concentration. It's always moving down that concentration gradient. So if a certain level of a molecule needs to be very high or very low inside the cell, we need to use energy. We need to use cellular energy to keep those levels where they should be, either very high or very low. In these examples, you can see we have our sodium. It must maintain very low levels inside the cell. We have our potassium. It must maintain high levels in the cell. And our calcium must maintain high levels inside the cell. So we need a different mechanism to keep high or low levels of some molecules inside the cell. We must use energy. In active transport, when those molecules need to maintain very high or very low levels in the cell, Active transport, we call that active transport, and that's when there are protein pumps that use energy and pump a molecule in or out of the cell. So we have our protein pumps. That is another thing that is embedded in that cell membrane, our protein pumps. So here you can see we just have a chart that is kind of laying it down in black and white about how molecules move across the membrane. It talks about diffusion, osmosis, facilitated diffusion, and active transport. All of these things are moving molecules in different ways. And all of them are passive, meaning they don't require energy, except our active transport. Our active transport requires that ATP, right? Remember that cellular energy, that ATP. So our active transport is moving molecules from low to high concentration. So we know our passive transport was talking about moving from high to low. This is moving from low to high. So this one is going to require energy. Now there's a couple different types of protein pumps, these movers we're going to talk about really briefly. So we have an antiport, first of all. This is the exchange port. So it transports one molecule from one side in exchange for one on another side, okay? So that's swapping them out. We have a symport, which carries two different ions or molecules in the same direction, two different ones. And we have a uniport, which carries only one specific ion or molecule. So these ones are specific to the molecule that they carry. They only carry one type of molecule or ion. So these all use active transport. They all need energy to move these larger polar molecules across the cell membrane. So when we're talking about our active transport, we have a couple of different types of pumps we can talk about. So we have our antiport, which is moving two different molecules in opposite directions. So, and remember, all of these use ATP. That is cellular energy. The mitochondria make cellular energy, and the scientific name for that is ATP, or adenosine triphosphate. I think it's ATP for short though. So this ATP, I will attach a video so you can watch the video. But in the video, you'll be able to see that this ATP, it's a little compound, a chemical compound. It will attach to these pumps, to all of these pumps, and it will break apart. So it will break off into ADP, adenosine diphosphate, and a phosphate. And that breaking apart releases energy, which allows these molecules to move through this port. So here we have an antiport. This has these molecules moving opposite directions at the same time. This symport is two molecules moving in or out. They're moving in the same direction, two molecules at the same time. And then we have our uniport, which is one molecule moving in or one molecule moving out at the same, at one time. And all of these use energy. They all use that ATP, that cellular energy. They break that ATP apart, release energy, and that is what allows these molecules to move from a low to a high concentration gradient into or out of the cell. So other than our protein pumps, we have a couple more types of active transport. So one of these is endocytosis. So endocytosis is where a molecule, a large molecule, wants to enter the cell. So what it does is it creates this pocket. It creates a bulge in that cell membrane, and that molecule then, that cell membrane pinches off around that molecule, and that cell membrane becomes a vesicle, okay? So in this example, a food particle, oh, my mouse has stopped working, so let's swap. A food particle has entered the cell. It is becoming surrounded by that cell membrane, and it's going to float through that cell, and eventually a lysosome, so those are our things that eat, the lysosome digests those food particles, and the products of digestion, they are released into the cell can use those. But the rest of it, the stuff it doesn't need, is going to exit the cell. So we call that exocytosis. So it enters via endocytosis. This process of being digested is called phagocytosis, which just means to eat on a cellular level, phagocytosis. And then those products, those waste products, are going to exit the cell through exocytosis. So that is another example of active transport. So here is a little animation of a vesicle fusing with the cell membrane. It's releasing waste products, or it might be releasing something that the cell made. So if it's secreting a chemical or a hormone that it made, the cell would use exocytosis. And that it just means that it's active transport of something exiting the cell, so something leaving the cell. And that happens because that vesicle fuses with that plasma membrane and those contents are released.